This is Unit 7 Notes, Part 2, DNA and RNA. This particular part is about DNA replication and RNA structure. We're going to begin with a video showing you basically what happens with replication, and then I'll explain to you what's going on in the video.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Okay, so DNA replication is the process of making identical copies of DNA. This happens when the cell is getting ready to divide and it needs to replicate the chromosome. So in order to make a copy, of course, the DNA molecule has to unwind. It's sort of like a zipper that just unzips. There's an enzyme called helicase that takes care of this unwinding process. And instead of just copying from one end directly to the other, because DNA is a very long molecule, actually this happens at several places along, a number of places along the DNA strand. So each place there's that helicase molecule that starts opening up the DNA, and then new nucleotides are added from the 3' end by the enzyme DNA polymerase, matching up complementary bases, complementary DNA nucleotides. And then the new nucleotides are connected to a chain by the enzyme ligase, which holds them together, basically like a stapler or like tape. Please remember when we talked the other day about DNA structure that the two strands of DNA run in opposite directions, depending on which direction the sugar molecules are located. One end runs from 3' to 5', the other runs from 5' to 3'. So if it starts unzipping at this end down here, the copying has to be done from 3 to 5. And so this part of the strand can copy all the way down very easily. But the other side has to be copied backwards. So when you unzip down a little bit, it has to copy just this part. And then when you unzip a little bit farther, it has to copy this part. And so what you end up with is a leading strand that's copied continuously and a lagging strand that has to be copied one piece at a time because it has to be copied backwards. The multiple places where this happens in the DNA strand are called replication forks. And when the process is finished, you will have two strands of DNA, each one that is half original strand and half new strand. Replication is what we call semi-conservative. That means that some of it is saved, half of it is saved. Each new DNA molecule is half original chain and half new chain. Now here's another video to help reinforce what we just discussed.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So DNA is copied like that. You end up, like I said, with two strands of DNA, each of which is half original strand and half new strand. Now, sometimes you have to also have a copy of just part of the DNA molecule to send messages to make things out of the nucleus. And that's when you end up making something called ribonucleic acid, or RNA. RNA is found both in the nucleus and in the cytoplasm. And there are three kinds of RNA that are named according to the functions that they have and the jobs that they do. Messenger RNA, or mRNA, is a strand that is complementary to or completing the DNA molecule. It copies the genetic message from the nucleus to the ribosome for a particular section of DNA. And its message is coded in a sequence of three bases on the messenger RNA molecule. The three bases are called a codon, and a codon codes for one of the 20 amino acids. The second kind of RNA is ribosomal RNA, also called rRNA, and it forms the bodies or the structures of the ribosomes along with some proteins. And of course, the function of ribosomal RNA, or the ribosome, is to make proteins. And the third kind of RNA is transfer RNA, or tRNA, and its job is to transfer the amino acids from the cytoplasm to the ribosome so that they can be put together in the correct order to make the proteins. RNA is also made of nucleotides, like DNA is, but the nucleotides are slightly different. They have ribose sugar rather than deoxyribose sugar. They still have a phosphate, and they also have one of four nitrogen bases, but the nitrogen bases are slightly different. RNA has adenine, cytosine, and guanine, like DNA does, but the fourth one, rather than thymine, is uracil. Thymine does not, uracil fits with or pairs with adenine like thymine does, but it fits better with the ribose sugar than thymine does. Each sequence of three bases in the messenger RNA makes a codon. On transfer RNA, there is an anticodon that is complementary to the messenger RNA. Another thing about RNA is that it is a single-stranded molecule rather than a double-stranded. Now, messenger RNA is a long, skinny piece of RNA that just has the copy of half of the DNA molecule, of a particular part of the DNA molecule that codes for a particular protein. Ribosomal RNA is part of the structure of the ribosome, of course, and transfer RNA has a really interesting, different kind of structure. Sometimes it's called a clover leaf or a key shape. I think it kind of looks like a T myself, but this is a two-dimensional picture rather than three-dimensional. And at one end of the molecule, there is the anticodon that is going to be the opposite or the complement of the codon in the messenger RNA. And at the other end, there's an amino acid. And each of the 20 amino acids has different kinds of transfer RNA with different anticodons that carry the amino acids to the ribosomes to make the proteins. This concludes the section of notes about DNA replication and RNA structure.